Thank you for your participation in today's event. The introductory segment of this series, Jeff Harford presented compelling reasons for researchers to consider near-infrared fluorescence for optical imaging of live mice. In this segment of the series, we will continue to build upon that theme by adding information on some of the commercially available optical probes and by providing a framework for you to use in the design and validation of your own optical probes. This presentation is designed to give you the tools and knowledge one needs to develop and validate your own near-infrared optical imaging agents. In our discussion today, we will be reviewing some of the principles outlined in the paper by Joy Kovar et al. On the development of near-infrared contrast agents for optical imaging of live mice, and showing examples of how we employed those approaches to the development of our own agents. Joy is online with us today as an expert panelist and will be able to take your questions as they come up during the presentation. We also highly encourage you to download a copy of this paper for your own use. It is a valuable resource to guide you in the development of your own near-IR optical agents. Licor is a leader in the development of optical imaging agents. These agents undergo rigorous development to ensure high specificity and binding capacity with excellent clearance properties. Currently, we have the IRDye 800CW EGF and RGD optical probes, IRDye bone tag, and IRDye 2-deoxyglucose. High affinity probes for a number of molecules can be developed by traditional design methods. In order to be effective, the agent must retain its biological activity while labeled with the fluor and reach the target in sufficient concentration and/or be retained there long enough to be visible at the time of imaging. Signal to background is also affected by absorption, distribution, metabolism, and excretion of the probe and the route of administration, and we will be addressing all of these factors later in the presentation. There are many considerations to take into account when choosing a suitable target for optical imaging. Many commercially available probes target cancer molecules or pathways, but investigators are certainly not confined to cancer when it comes to target selection. The benchmark criteria we will talk about in our evaluation of optical probes is the signal to background or signal to noise ratio and the importance of making good target selection to the overall determination of the quality of your probe. Ideally, one would like to target to be specific, abundant, disease-related, easy to probe, and easy to detect and visualize in vivo. Through the application of a systematic approach to optical probe design, one can obtain the answers to these questions and more. In general, the idea of systematic development of optical agents begins with the labeling of the ligand, conducting in vitro specificity assays, conducting in vivo clearance and specificity studies, and finally validating specificity ex vivo with organ imaging and histology. As I often say, labeling a molecule with dye is relatively easy, but making a validated optical probe is considerably harder. We at Licor strive to take every means available to ensure that we are providing quality optical imaging agents for you to use. We will illustrate how this paradigm has been employed with two different examples developed internally and made commercially available by Licor. The first probe development project we will discuss is that of IRDye 800CW EGF. The IRDye 800CW EGF optical probe is an excellent agent for cancer research. This agent will target tumors that overexpress EGF receptors. Since many tumors overexpress EGF receptors, this agent works very well in tumor progression studies. The image shown here is of an NOD skid mouse subcutaneously implanted with prostate tumor cells. The optical imaging agent is IRDye 800CW EGF injected intravenously, and the animal imaged 96 hours post-injection. The tumor labeling is pseudo-colored to indicate fluorescence intensity at 800 nanometers. Images were captured with the Pearl imager. Some of our initial objectives when developing this optical agent were to evaluate the binding activity of the ligand in vitro, confirm its specificity in vitro and in vivo, ensure good binding capacity and clearance, and validate the tumor tissue specificity with histology. An important consideration to remember, especially when labeling small molecules, peptides, and antibody fragments, is that coupling them to a very large fluorescent dye molecule might change the physical properties of the ligand enough to alter or eliminate its activity at or recognition of the target. The Odyssey and Pearl workstation will provide you a faster and more cost-effective means for agent development by letting you use one set of tools for all of your in vitro and in vivo validation needs. Using the Odyssey, one can first test the specificity of the optical agent using a cell-based assay we call the in-cell Western, or ICW. In this study, we demonstrate the dose-dependent labeling with IRDye 800CW EGF probe and two different cell lines that are known to express EGF receptors, shown in panel A, and the lack of binding of IRDye 800CW carboxylate, or free dye, to the same cell lines, shown in panel B. In panel C and D, we show that the binding of the IRDye labeled EGF could be blocked in a dose-dependent fashion using either pre-incubation with an antibody to the EGF receptor or unlabeled EGF. The results of this simple in vitro study accomplished several critical components in our quest for a validated target and optical probe. The first is that the coupling of the dye itself to the ligand did not render the probe inactive. The dye itself does not bind to the target, and that the binding of the probe to the target can be competitively inhibited. Another important consideration for optical probe activity, particularly when labeling peptides and recombinant proteins, is the source of the unlabeled molecule. Ligand evaluated recombinant EGF from 5 different suppliers before deciding on one we chose. This was done by labeling each different EGF molecule to the same specific activity and then conducting ligand binding assays on cells overexpressing EGF receptors, again using the in-cell Western assay. The results were dramatic and demonstrated to us the importance of being careful in our selection of suppliers for unlabeled EGF. The impact of this in vivo is seen in the next slide. Here you see the impact of using a ligand with poor binding activity. The image in panel A depicts strong binding with the dye-labeled probe from source 4, which had the highest binding activity in vitro, and the tumor is brightly visible. In panel B, the tumor is barely detectable when labeled with IRDye 800CW EGF from supplier 1, which had low binding activity in vitro. Ligand can also supply you with our 800CW dye in carboxylate form, which will be unreactive in aqueous solution. The free dye itself can and should be evaluated for clearance to ensure that it does not bind non-specifically to the model being used, and also to function as a baseline for later comparisons. The labeled optical agent should also be tested in a negative control animal, for example, a non-tumor bearing mouse. This will demonstrate how the agent performs in vivo in the absence of the target, and also will help to establish an initial time course for clearance of the unbound probe. In this slide, we show the time course for clearance of IRDye 800CW EGF from a non-tumor bearing mouse. A SCID mouse was injected with 1 nanomole of the EGF conjugated optical agent intravenously and imaged at the indicated time points over a 24-hour period. Analysis of the abdominal region showed that greater than 90% of the fluorescence disappeared in 24 hours, and by 48 hours, the probe had cleared completely. One can also evaluate the impact of route of administration on clearance of unbound probe in the same way. Many optical probes do not perform well when given IP. Finally, we tested the IRDye labeled EGF optical probe in tumor-bearing animals to establish an optimal time point for post-injection imaging. Keeping in mind that our benchmark value for comparison is the signal to background, or in this case, tumor to background ratio. We want to optimize the time for elimination of unbound probe while maximizing the accumulation and retention of the dye-labeled probe at the target. In this case, that time point was demonstrated to be 72 hours post-injection for the IRDye 800CW EGF optical probe from Ligand. The final step in our evaluation of our EGF optical probe was to demonstrate in vivo specificity. The tumors were excised, cryosectioned, and the resulting slides scanned on the Odyssey to determine tissue localization of the labeled EGF probe. Tumors that received no 800 dye labeled probe show no signal at 800 nanometers, pseudo-colored green. Either subcutaneous or orthotopically transplanted tumors from animals that were injected with IRDye 800CW EGF both showed tumor-specific labeling that could be competitively inhibited in vivo by pre-dosing the animals with C225 antibody to the EGF receptor. For us, this was conclusive evidence to demonstrate that our IRDye 800CW EGF was a validated optical probe for tumors expressing EGF receptors. But was the EGF receptor a good target for optical imaging? That is another question entirely. Since not all tumors express the EGF receptor abundantly, it was clear to us at Ligand that we needed to provide additional solutions for optical imaging of tumors in vivo. The second such probe that we have developed that I am going to discuss today is our newly launched IRDye 800CW RGD, which specifically targets integrin receptors. Integrins are cell surface heterodimeric glycoproteins involved in cell-to-cell and cell-to-matrix interactions. The recognition motif, RGD, or arginine glycine aspartate, is a tripeptide sequence used to bind integrin receptors, including the alpha-5 beta-3. Interest in using a labeled RGD peptide ligand for the study and or monitoring of diseases related to the alpha-5 beta-3 receptor overexpression is increasing. Several groups have labeled RGD successfully with fluorescent dyes for in vitro and in vivo imaging. Integrins have been demonstrated to be involved in tumor growth, tumor invasiveness, metastasis, and tumor-induced angiogenesis. The alpha-5 beta-3 integrin receptor subtype is overexpressed in tumors undergoing angiogenesis, such as melanoma, glioma, prostate, and breast cancers, while weakly expressed on resting cells and most normal tissues. These characteristics fulfill several of the criteria established earlier for good target selection: abundance, tumor specificity, and biological relevance. After successfully labeling the RGD peptide with IRDye 800CW, the next step in the development of this optical probe was to confirm its binding affinity and specificity to the intended target in vitro. This was easily accomplished with a cell-based assay such as on-cell westerns, again using the Odyssey. One of the studies conducted to demonstrate the specificity of IRDye 800CW RGD for integrin receptors is depicted here. In this study, a nonspecific peptide, RAD, or arginine alanine aspartate, was also labeled and used to evaluate binding. As expected, the nonspecific RAD peptide did not bind to the same degree as did RGD, confirming the IRDye 800CW labeled RGD peptide is specific to the cell line expressing alpha-5 beta-3 integrin. Use of multiple tumor cell lines will provide a level of assurance that the agent is broadly applicable. In the data shown here, we have interrogated 2 prostate cell lines. Binding affinities for both cell lines are shown at the top of the graph, whereas a competitive blocking study is presented at the bottom. In this assay, unlabeled RAD and RGD were used to compete for binding sites. Specificity is confirmed when RAD fails to block binding of IRDye 800CW RGD, while unlabeled RGD successfully inhibits binding. As with the development of the EGF optical probe, the next step in the development of the RGD probe was to determine the optimal pharmacodynamic properties of the probe and to confirm specificity in vivo. This slide depicts 3 different doses of the RGD optical probe administered to animals bearing U87 subcutaneous tumors on the right hip. The images were all acquired 24 hours post-injection by the pearl imager. In this case, at 24 hours post-injection, the RGD optical probe did not give sufficient tumor to background signal until 1 nanomole dose. Identifying the optimal imaging time point, or pinpointing the time when signal-to-noise ratio is at its peak, allows the researcher to streamline the imaging process. Here, an animal bearing 2 tumors, left and right hip, was injected with IRDye 800CW RGD and then serial imaged over the next 96 hours. Tumor signal-to-noise ratios were determined in the PearlCam software and plotted. Note tumors from different origin may differ in clearance, reflecting a slight shift in the optimal imaging time point. However, signal-to-noise ratio peaks were reached between 24 and 48 hours post-injection. This gives the researcher a window to choose an imaging time point that best suits their study parameters. Once a time point is chosen, it should be maintained throughout the study. Data in all early phases of the development has shown the agent to be specific. However, the ultimate test involves competitive challenge in vivo. When signal is measured for both tumor models, approximately 70% blocking was achieved for U87. Interestingly, A431 tumors did not exhibit the same level of blocking, approximately 5%, which demonstrates the variability between tumor models. A431 tumors do tend to get necrotic very early, which may cause some pooling to occur. This pooling may have masked the blocking effect. As with the EGF optical probe, the final step with the RGD optical probe was to excise the tumor, prepare tissue sections, and examine them further. Tumors from the previous study were excised and frozen sections prepared. Analysis of 800 nanometer fluorescence intensities per area showed both tumors were successfully blocked when unlabeled RGD preceded the probe injection. These data confirm that the specificity we noted in vitro was maintained in vivo. Finally, in a separate study done to examine the cellular location of the dye after the labeled probe had bound to the cells, we employed fluorescence deconvolution microscopy to evaluate the location of the optical agent more closely. In the case of the IRDye 800CW RGD, we see that the dye has been internalized as demonstrated by the fluorescence, pseudo-colored green, detected in the cytoplasm of these lung carcinoma cells. The nucleus of each cell has been stained with Cytox green and is pseudo-colored blue. Another benefit of Lycor IR dyes is their suitability for fluorescence microscopy. Two additional optical probes from Lycor that we will not explore in as great a detail today are the IRDye 800CW 2DG and bone tag. IRDye 800CW 2DG is a very exciting imaging addition to our optical agents for cancer research. This probe will target cancer cells with elevated rates of glycolysis and will be the subject of an additional webinar at a later date. The IRDye bone tag provides an exceptional structural agent. This agent is available in both IRDye 680 and IRDye 800. When imaging tumors, The IRDye 680 may be used in conjunction with IRDye 800CW tumor agents for dual targeting experiments. This agent incorporates into the bone and can be detected for several weeks post-injection for longitudinal imaging. One significant advantage of the Pearl imager is that it can excite and detect fluorescence at both of these wavelengths in a multiplexed way without having to perform spectral unmixing of the signals. Vizen Medical also offers a suite of near-infrared in vivo optical imaging agents. One important distinction between the two sets of probes is the 750 nanometer excitation wavelength of the Vizen dye. While 750 nanometer wavelength is better than 680, it is not optimal for fluorescence imaging in vivo, and dyes that emit at this wavelength do not have the same physical and optical properties as the IRDye 800CW. The 800 nanometer wavelength is ideal for in vivo imaging, and the IRDye 800CW has superior physical and optical properties. The Pearl imager is the only commercially available small animal in vivo imaging optical platform specifically designed and matched to excite and detect IRDye 800CW. Well, what if you have discovered the next abundant biologically important and tissue-specific biomarker for cancer or something else, and we don't have an optical probe available for it yet? Well, call me immediately before you do anything else. Or you might consider ordering a dye labeling kit from us, or ordering the dye itself and labeling the molecule yourself. Lycor has labeling kits for large or small molecular weight proteins and kits for labeling smaller batches for 800CW, 700DX, and 680 dyes. Bulk dye is also available in 0.5 milligram and 5 milligram packs with either NHS ester or maleimide reactive groups for coupling. Be sure to employ all the necessary steps to validation outlined here today to improve your chances for successfully generating your own in vivo optical imaging probe. So in summary, what we've tried to demonstrate today is what can be targeted for in vivo optical imaging. In general, targets that are abundant, relevant, and tissue-specific make the best targets. How to target, to test labeled probes in vitro first before proceeding to in vivo studies. The importance of dosage and clearance in determining optical imaging time points, and the importance of using histology to validate your optical probe performance in vivo. That is the end of the seminar for today, and we will transition now into the live question and answer period.